phone. You want him? All right, we're going to get going. So welcome, everybody, and thank you for being here for the ASN Vice Presidential Symposium on reviewing the role of genetic variation in adaptation. My name is Deepa Agashe. I'm from Bangalore, India, and it is really an honor to be able to organize this symposium and have a set of really wonderful talks from a bunch of invited speakers, as well as contributed talks from people who submitted abstracts, and their work was completely spot on and aligned with the symposium. So I'm really excited about the set of talks that you're going to hear today as part of the symposium. What I'm going to do in my sort of half-hour slot at the beginning is to set some context and explain to you what I have been thinking about when choosing this topic and the work that you will listen to in today's symposium. So in contrast to the kind of work that you heard from Judy just before, which I think exemplifies what ASN stands for in many ways, we're going to shift gears a little bit, move away from a lot of ecological, macro-organismal work to start off with much more a molecular focus on trying to understand genetic variation and sort of get understanding of what is its impact on evolutionary dynamics. I should have looked at clicker. Okay. So as you all know, genetic variation is a key parameter in ecology and evolution. I do not have to tell this audience this fact. And the reason it's a key parameter is not just because of what we know from basic population genetic theory, for example, that genetic variation is the underlying raw material on which selection acts. The repercussions of genetic variation extend far beyond the specific population and the specific organism that we're interested in. We have ecosystem-level impacts as well that are very clear and demonstrated in different kinds of habitats. So here is an example from a very nice review paper where you see that in these trees that are keystone species in this environment, in this habitat, genetic diversity in these trees at the genotype level across individual trees influences how beavers use these trees. And depending on the tannin content of the different trees, that has all of these cascading effects in different aspects of this ecosystem. So there are impacts of genetic diversity that range well beyond the specific species or population. You also all know that understanding genetic variation is important for practical applications. Here's an example from crops, different crop species that have benefited from a deeper understanding that we have developed over the last few decades of research on which kinds of genetic variants are important for giving rise to the traits that make these crops viable crops and increase productivity. And examples here include the shape or the size of the seeds, which we have, of course, also selected upon in the process of domestication. Another important application in some sense is the issue of conservation of species. We know, again, that the capacity of species to adapt to environmental change, whether it is anthropogenic or not, is going to depend on the ability of the population to adapt, again, that relies on genetic variation being available for selection to act upon. So given all of this background and the vast amount of work that we already have in the field on this subject, what is the need to revisit the role of genetic variation? And when I say revisit, I don't mean let's turn everything upside down. I mean that there are a lot of interesting details that have emerged in the last few years that we need to really start incorporating much better. So here's an example of some recent advances that I think really call for a more nuanced understanding of how genetic diversity really matters in evolutionary and ecological frameworks. First, we have much more explicit molecular mechanisms, an understanding of molecular mechanisms that generate variation and the rates at which these variation is generated. And this molecular accounting would help us understand much better how genetic diversity can impact evolutionary dynamics. We also have much better quantification of the relationship between genotype and phenotype and fitness, which are, of course, very, very key parameters that help us, again, understand how genetic diversity is going to impact evolution or ecology. And finally, there are much larger data sets now and detailed data sets on the genomics of adaptation in natural populations, because ultimately what we're all trying to understand is how evolution works in nature. And we can't do that if we are not able to connect what we are seeing in the lab or in theory to what happens outside in real populations. So having data sets on genomics of adaptation in natural populations allows us to test some of these ideas that come up from the work that we do in silico or in the laboratory. So here are three major questions that I would like to highlight, which are going to be the subject of today's symposium. One is, how does mutation rate and bias alter evolution? What is the phenotypic effect of mutations? And how are old versus new mutations sorted during adaptation? Some of these are very old questions, and we simply have much better data and much better understanding. But some of these are questions that have emerged as much, much more important questions than we used to believe in the past. So let's start with the first one. What I'm going to do here is give you a brief introduction to some background for each of these questions, and then highlight the talks that are to come in the symposium and how they connect with these different questions. This is a very nice figure I like from Ruth Hirschberg's review paper a few years ago on how mutation bias and selection should both determine what kind of substitutions are we going to observe in populations that we're interested in observing. So if you look at the process of selection as the sieve, under normal levels of selection, where there is where selection is going to filter out some mutations, you may have mutational input be different, right? So different types of mutations may be more or less likely to occur in a population. The selection sieve is then going to sort through those and, let's say, not allow these mutations to persist so much. The blue ones are gone, and you have only a smaller subset of substitutions that you will observe in the population because the rest have been thrown out by selection. But nonetheless, there is still an impact of which kinds of mutations feed into this whole system to begin with, right? So if you don't have blue mutations at all, you will, of course, never see them. But if you have a lot of red mutations, you may be more likely to see them on this end after selection has acted. And that is what I mean by mutational bias, being an important factor that we have not considered as much in the past. If, on the other hand, you have relaxed selection, then the sieve is much more porous. It lets much more of the natural mutations that have occurred in the past, it lets most of them through, and then you do see that the substitutions reflect the mutational process much, much better at the end of it. So in today's symposia, you're going to hear after me from Arlyn Stoltzfus, who's going to tell us about the historical perspective and new advances in understanding the evolutionary impacts of mutation biases. And so I encourage that you stay for that talk because it's going to be really interesting. And we have a short talk later by Ahmed, and he's going to tell us about stress-induced mutation biases in Chlamydomonas. And that's such a great example of, I think, a nice way to show you that, well, here's a new system now where we understand the mutational biases, quantify them, and then we can go on to ask what impact they have on selection. Now, I just want to give a bit of a primer on mutation biases because I want to tell you a little bit about work that we have done in my lab in the last few years, which has been really exciting. So mutation biases come in many flavors. You may have transitions or transversions as one way to classify mutations. Transitions go from purines to purines or pyrimidines to pyrimidines. Transversions are everything else. And there are many more transversions, as you'll see here, right? So in a situation where there's no mutational bias at all, you'd expect that 2/3 of all mutations should be transversions, and that's what we should sort of observe before selection acts on mutations. But in fact, most organisms show a transition bias, And this is true even in mutation accumulation studies. When we try and minimize the role of selection, we still see a transition bias. And this is of varying strength. Now, why there's an underlying transition bias remains kind of unclear still. But what is more fascinating and interesting for evolutionary purposes is to understand why the bias is of different strengths in different organisms, right? We know that mutation bias has many important effects. This is work that has really come up in the last few decades, very nicely showing that it can determine the genetic basis of adaptation. It can drive parallel evolution. So at least a substantial fraction of times when we observe parallel evolution at the genetic level, it seems to be driven by the mutational biases that exist in the first place. So if some mutations are more likely to occur, they're also more likely to be favored by selection eventually, because that's the pool of variance that goes in, right? So this can be driven at least sometimes by mutational biases. And this is ultimately important to predict drug resistance mutations as well, so has practical importance and applications. What I want to show you briefly is some work that we've done recently to ask what is the impact of mutational biases on fitness effects, right? And so we've done these experiments in E. coli populations, and we measured in wild-type E. coli as well as a mutated version of E. coli where we deleted some DNA repair, allowing a different kind of mutation bias to exist. So the wild-type E. coli has a strong bias towards or a reasonable bias towards transitions, like most species that I told you about earlier, whereas this mutant that we made is transversion biased. And we simply asked how many of the mutations that we observe, randomly chosen, are beneficial or deleterious and so on, right? And we see very, very interesting differences here. What I'm showing you here is the number of mutations that are consistently beneficial or consistently deleterious in a large number of environments where we tested the impact of making these mutations. And what you see is in the wild type, shown here in this green, beneficial mutations tend to be beneficial in few environments. So if we identify a beneficial mutation, let's say in a particular glucose medium, it is not likely to be also beneficial in other media where we test this. Whereas with deleterious mutations, any given deleterious mutation in one environment is also likely to be deleterious in at least 5 other environments. That's sort of what we see very often. In contrast, for this mutant, which is transversion biased, many more mutations are beneficial in a larger number of environments, whereas mutations tend to be deleterious in a very few number of environments, right? So this pattern is completely flipped. And this is one of the first demonstrations that if you change the pool of mutations coming in, you are going to change what is the impact of these mutations going forward during adaptation. So what we're suggesting based on this work is if you think of a fitness landscape as a cartoon like this, where you have transversion biases, maybe some other mutational biases, like such as AT and GC bias, and you have fitness peaks. If you are transitioning to a different mutation bias, you are actually likely to be able to access fitness peaks which you were not able to access before, right? And this might be why mutation biases may persist and may be shifting around across evolutionary time. So this work is up on bioRxiv and was done in collaboration with Lindy, who's also another speaker in this symposium. And I want to highlight that we predict that when you have these complete flip-flopping of the mutational bias, which should change what is the pool of variance which is available for selection, you might have slower Muller's ratchet. So when populations are evolving under genetic drift, they should accumulate mutations that are deleterious, but that process should slow down when you have a shift in mutation biases. You may also have faster adaptation when populations are evolving under selection, and the mutation rate and spectrum should jointly impact the dynamics of populations. And going forward, one of the other talks in the symposium by Lindy is going to explain a little bit more about the simulation work that was done for this work, as well as a follow-up body of work that Lindy has been doing to ask, well, what happens if mutation rate and bias both are shifting at the same time and both are allowed to evolve? How does that work? So you'll hear from Lindy, and you will also hear from Adi Leavnat, who's going to talk about mutation rate variation across human hemoglobin gene. And so this is, again, another very nice example showing that the rate variation within a particular gene, which is under strong selection, can also have potential impacts for adaptation in a real-life scenario. Okay, so I want to move on to my second question, which is what is the phenotypic and fitness effect of mutations in populations? And one of these really interesting things that I think has emerged in the last few decades, which we didn't have before or understanding about before, is that synonymous mutations are not really neutral fully. And so what I'm using here is a schematic, like a cartoon summary from a review that I did recently, showing you what kind of direct evidence do we have to make a statement like this, right? And I think all of you already know that synonymous mutations are often used as the baseline for neutrality in analyses of selection at the genome level, at the genetic level. And so it's important to know whether this baseline is a valid baseline and when should we reconsider it and why, under what circumstances. So here's one example of direct evidence. Well, it's not an example, it's a schematic. What kind of evidence do we have when people have made random mutations in focal genes of interest in, let's say, microbial populations and measured the impact of these mutations on fitness or phenotype of interest? They find that synonymous changes, shown here in pink, often do not have a significantly different impact on fitness compared to non-synonymous mutations, right? And this is all with respect to the ancestral variant. Similarly, if you plot distributions of fitness effects, which are histograms of fitness effects, you again find that for synonymous mutations, these distributions do not look very different from the distributions observed for non-synonymous effects, again suggesting that thinking of synonymous mutations as being kind of neutral baselines is perhaps not such a great idea. And finally, there are also very good examples, including some from work in my lab, showing that adaptive mutations that evolve under selection in the laboratory under experimental evolution can include a bunch of synonymous changes, and we can show by cloning these mutations back into the ancestor that that is the one synonymous mutation that has an enormous impact on fitness, right? So this is one kind of new evidence that has come up that forces us to think again about what kind of genetic variation should be contributing or could be contributing to adaptation. And more generally, we have a much better handle in the last few decades on the distribution of fitness effects of mutations in many, many different kinds of organisms under different environments. What you see here is a plot showing how the distribution of fitness effects differs for the same set of mutations across different environments. And so you see it's generally bimodal. It's not unimodal, as we have thought earlier in the past so much. But the nature of that distribution does vary quite a lot depending only on the environment, right? So this is expected from base theory, but it's nice to see data that show this very clearly. So following up on this, though, what we want to really understand is how these kinds of effects observed at the single mutation level in laboratory populations, do they actually work and translate and help us understand something about natural populations? And the next two talks in this symposium would address this. So Katrina McWegan is going to talk about mutational effects for swimming speed in zebrafish, who are being forced to swim in a natural-like condition in the laboratory, perhaps. And so how does this differ across environments? And then Melissa is going to tell us about adaptation in environmental gradients via regulatory variation in sea urchin populations across, the western coast of the US. So these are going to be great examples of understanding what kind of genetic variation is responsible for adaptation of different environmental conditions in natural populations. Okay, and then finally, the third question that I want to address, how are old versus new mutations sorted during adaptation? I'm not going to touch upon this in too much extent in terms of background because that is not my cup of tea so much, and I have never worked on this. But what I want to summarize in one slide from a lot of work that is going on this topic is that selection produces distinct patterns of genetic variation across the genome, and this is a slide from a very nice review from a few years ago, which shows what happens to heterozygosity across the genome of populations selected for development rate or control populations shown in blue. And you have very distinct patterns of heterozygosity, which you do expect. But then, of course, there are lots of nuances and kinks in this, right? So it doesn't always work out the same way. There are differences in depending on what kind of selection is acting on populations, which kind of selection pressures are acting, how many, and so on. And so you will hear 2 more talks in the symposium, one by Chengde Li, who's going to tell you about the fate of genetic variation under climate change, focusing on populations of banana plants. And finally, Jae-Young Choi is going to talk about the role of ancestral variation in a woody plant adaptive radiation in Hawaii. And so that's kind of where the symposium is going. I hope you can see that there's a breadth of different questions we can start understanding much better. The effect and fate of variation depends on mutational processes in addition to selection and demography. Assigning neutrality versus selection at the genetic level, once we start becoming explicit about what we mean by genetic variation exactly, is a bit fraught. It's not as sort of simple and straightforward as we would wish it to be. And there's much to learn about genetic variation in natural populations that we are slowly building up to now. So with that, I'm going to stop and thank the ASN for having me here and giving me this platform to be able to organize this symposium. I'm really excited about the talks that are coming up. I'm also thankful to the organizers of Evolution, the meeting here. It has not been easy, but I've really enjoyed myself so far and look forward to the rest of it. I'd like to acknowledge my home institution, the National Center for Biological Sciences in Bangalore, which has been a great place to work, and my major funding support from India Alliance. And finally, my lab, without whom nothing is possible anyway. So it's a great group to work with, and I'm happy to take any questions. I have no idea how much time is left, but questions or comments, actually. Yeah, I'm sorry. I think so the question is, was there a shift in the number or fraction of beneficial versus deleterious mutations in the transversion bias mutant versus not? And yes, I think I had that up there, but I forgot to mention it. So I think about 28% of mutations in the wild type, which is transition biased, were beneficial. And that number increases to more than 35-40% in the mutant, which is transversion biased. All right, if there are no further questions, we'll close and then wait for the chimes, and then the next speaker is going to be Arlene.